Good evening, NC team, junior doctors, I future doctors. Shall we start this stream now? For 30 minutes, only for 30 minutes. I hope before starting the stream, I want to check you that whether you have studied, whether you revised yesterday's neural log series day 9. So shall we start with the recap? If you missed it, no problem. We'll do it right now. I want you to quickly pick your pen and paper for today's series. So today we'll have the recap first. Block 1, prokaryote versus eukaryote. Under this we saw the ribosomes. It is a non-membrane bound organelle. It is a universal organelle because it is present both in the prokaryote and in the eukaryote. Next, it is made up of two subunits, one larger and one smaller in both prokaryote and eukaryote. In prokaryote, it is of 70s type. In eukaryote, it is of 80s type. That is 50 plus 30, the larger and the smaller subunit. Here 60 larger and 40 smaller. Okay. Next, what is the function of ribosomes? Students, I want you to type it in the comments. What is the function of ribosomes quickly? What is the function of ribosomes? Next, what are the organelles present in the, what are the organelles which contains ribosomes? Okay, usually ribosomes are present in the cytoplasm, isn't it? Cytoplasm of the cell. Where is the ribosome seen in the prokaryote? This is today's question. Okay, today's quiz is the ribosome. Where is the ribosome present in a prokaryotic cell? Student, this is the, I asked you what is the function of ribosome. The first question, this is the second question. Ribosomes are present in the cytoplasm of the cell. It is present in the cytoplasm of the eukaryotic cell. In the prokaryote, where will you see the ribosomes? This is the second question. Quickly type the answer in the comments. Then in the cytoplasm, what are the other organelles which contain ribosomes? Mitochondria, chloroplast. These two are semi-autonomous organelles. I am repeating it because very important, semi-autonomous organelles. Then in the rough endoplasmic reticulum. What is the special characteristic of rough endoplasmic reticulum? They contain ribosomes. Okay, the granular structures. Suppose if there is no ribosome, what do you call it as? Smooth endoplasmic reticulum, SCR, smooth endoplasmic reticulum. Now, what is the function of rough endoplasmic reticulum and what the function of smooth endoplasmic reticulum? Quickly type the answer in the comments, students. Students, are you following me up? Are you following me? I want you to write yes in the comments because I want whether you are present here mentally, physically, completely focusing on the current stream. Okay, this is the block 2. Under this we saw nucleus, that is chromatin versus chromosome. That is, both are the same but the appearance differs. Okay, they are made up, both are nucleoprotein complexes. The composition of that is DNA plus histones. They are basic proteins, non-histone proteins, and RNA, very very important. Next, when can you see the chromatin and when can you see the chromosome in a cell? In the interphase, that is in the resting phase, you have nucleus. Inside the nucleus, the loose network of nucleoprotein is called as chromatin. Who named this? Can you tell me the name of the scientist? The question of the day. The question of the day. Who named it as chromatin and what dye he used? Okay. Next, in the dividing cell only you can see chromosomes. Okay, visible only in the dividing cells. These chromatin, loosely interspersed chromatin, becomes tightly compact. Okay, it becomes tightly packed to form the chromosome in the dividing cell. So this you can see in the cell division. Next, what you see, based on the centromere position. What is centromere, students? Type the answer quickly in the comments. Okay, what is the meaning of centromere? The constriction of the chromosome, you call it as centromere. So based on the centromere position, you divide the chromosome into metacentric, submetacentric, acrocentric and telocentric. If it is exactly in the middle, you have two equal arms, you call it as metacentric, and it's like the letter shape V. Okay, next you have submetacentric. In this, it is slightly away from the middle, forming one short and one long arm. Okay, it looks like L shape. Next is acrocentric. In this, it is close to the end but not at the end. One extremely short, this one, and very long. This is J shape. Next is telocentric, where the chromosome, where the centromere is at the terminal, the last, the dead end. Okay, it looks like I shape or rod like. This one we saw yesterday. How many of you are able to recollect how many blocks, how many code words are you able to recollect? If you are able to recollect, type logged. Type logged in the comments, students. Type logged. Type the word logged in comments. I want you to type the word logged in the comments. Okay. And today we are going to see day 10 new neural log series. First is the polyribosome or polysomes. Polyribosomes or polysomes. This is the other word. See, Neet can ask you either as polyribosomes or polysomes. The word, see, always split the word. Poly plus ribosomes. Poly means many. Ribosomes. So, many number of ribosomes. Okay, from this itself you can understand. Okay, what is the meaning of polymer? Chemistry, monomer. Do you know the meaning of these words? So poly means, this word poly stands for many. Okay. So polyribosomes means many number of ribosomes. It is also called as polysomes. It is very important. Now it is very easy to remember. P for P. It is seen in the prokaryotic cell. It is seen in the prokaryotic cell. So polyribosomes or polysomes is the code word. They can ask you like this. They are present in the cytoplasm of the prokaryotes. What are those actually? There are several ribosomes, that is many number of ribosomes in a single mRNA strand. I'll draw and show you now. In a single mRNA strand which forms a chain, you call it as polysomes. What is the function of polysomes? Translation of mRNA into proteins. They translate mRNA into proteins. Okay, now I'll show you. See, one mRNA strand. Okay, I'm going to draw a strand. Imagine this is the thread. Okay, a thread, a single thread. How you see? Have you seen beads? Okay, mala beads. It will be like this. This is one design. You have continuous beads. Sometimes there will be gap between the beads. Imagine like this. A bead with a distance between them. Each bead has distance between them. Okay. Same way this thread is the mRNA strand. Single mRNA. The same thread from here to here. It is the same thread. And imagine these beads are ribosomes. They are what? Ribosomes. The larger unit and smaller unit. Okay, so several ribosomes, many like this, many number of ribosomes like this goes. Okay, so several ribosomes attached to this one mRNA chain, you call this chain as polysomes. Understood? Polysomes or polyribosomes. Okay, now what is the function of this polyribosome? Translate this mRNA to protein. Because you know this is seen in the prokaryotic cell. For the prokaryotes, do the prokaryotes have nucleus, students? There is no nucleus, no nuclear membrane. Now I have a question for you. Where does the translation takes place in an eukaryotic cell? In an eukaryotic cell. Where does the translation takes place in the eukaryotic cell? Type the answer in the comments, students. Type the answer in the comments quickly. So now shall we write this code words? If they have given polyribosome or polysome, search for these code words. Okay, first one is this. First one is this. This is very important. So we'll start with this. Several ribosomes, one strand of mRNA, very very important. One strand of mRNA. I told you, no, single chain, single thread with the beads. They are the ribosomes. Okay, so several ribosomes. Again, we'll repeat it one more time. Students, are you following me up? Students, if you follow me, type yes in the comments. Students, several ribosomes in one mRNA strand. What is the function? This is second. This is the first code word. Second, this. Okay, what is the function of polysome or polyribosome? Translation of mRNA into protein, because the prokaryote does not have a well developed organelle, no nucleus, no nuclear membrane. Okay, so polysomes does the function of translation of mRNA into proteins in the prokaryotic cell. Okay, in the prokaryotic cell. We saw already, isn't it? Prokaryotes lead a very simple lifestyle, minimal lifestyle. Remember minimalism. Okay, next, this is the, sometimes this can be given as the code word, that is, they are seen in the cytoplasm of the prokaryotes. Polyribosomes are seen in the seed. Because ribosomes, you may confuse it, you may choose endoplasmic reticulum if they are given choice, but you have to remember there is no endoplasmic reticulum in the prokaryote. Okay, but for that, first you have to understand that polyribosomes are seen only in the prokaryote. I told you, no, P for prokaryotes. Okay, P for prokaryotes. Now they are seen in the cytoplasm of the prokaryotes. Students, do you get the points? Code words, did you lock it in your gray cells? If yes means type it in the comments, students. Type it. Next, second is, we have seen under the block 1, what did we see? Polyribosomes or polysomes. The first code word is several ribosomes attached to single mRNA strand. This chain is called as polysome. The function of polysomes is to translate mRNA into protein. They are seen in the cytoplasm of the prokaryotes. Fine. Next, you have chromatophores. So what do you know about chromatophores, students? What do you know about chromatophores? I think I hope you have studied the first chapters. Did you study about photosynthetic autotrophs? Photosynthetic autotrophs. In the beginning, okay, photosynthetic autotrophs. I am talking about chromatophores and photosynthetic autotrophs. Are you able to connect both? Are you able to connect both? This is in the cell chapter. This is in the beginning, okay, when they are describing about bacteria, isn't it, about the kingdom. Are you able to connect both? If you able to do the critical reasoning, yesterday I told you, analytical thinking, you type the answer for that. Chromatophores are very important. This is given in cell chapter, but it is also interlinked to the kingdom, okay, the subtopic kingdom, where you can read the word photosynthetic autotrophs. I want you to go take your NCERT book and try to match this with this today. Okay, today you are going to do this, that is cell chapter and the kingdom, subtopic kingdom. Chromatophores are seen in cyanobacteria. Now the question comes, we already saw this. What is the other name of cyanobacteria, students? What is the other name of cyanobacteria? We saw it in bacteria, examples of bacteria. It is also called as blue-green algae. Students, if you are not able to catch me up, you can type it in the comment section which part you are not able to get into, because I'll repeat it in the other class or make it as a shorts or a video. We have dedicated only 30 minutes for this, so I don't want to get deviated. I'm going ahead, but that doesn't mean that you should not express, or if you have any doubts or you want me to do any new chapter, you can definitely leave it in the comment section. Once the session gets completed, I'll go through it and I'll definitely make a video out of that, or I'll give you a reply in the next class. I mean tomorrow. Okay. Next, chromatophores, they are seen in cyanobacteria. Cyanobacteria is also called as blue-green algae. I told one trap related to this. Okay, this is not an algae, not a eukaryote plant. This is a prokaryotic bacteria. We already saw. If you are able to remember, type S in the comment section. Next, chromatophores are seen in the cyanobacteria. What are these chromatophores? They are extensions of the plasma membrane which contains pigments. What is the pigment? Chlorophyll A. Because of this pigment, it is able to synthesize its own food. That is why they are called as photosynthetic autotrophs. Now, are you able to understand the reason behind this? Chromatophores are plasma membrane extensions which contains the pigments. Okay, the pigment is chlorophyll A. This chromatophores are seen in cyanobacteria. That is why cyanobacteria is able to synthesize its own food. That is why it is called as photosynthetic autotroph. They can ask this as AR type of question. When I studied, I didn't get this, but when I am explaining you, I myself finding out this can be asked as an AR type of question. What did you think, students? This can be asked as AR type, right? See, cyanobacteria are able to synthesize their own food because they are called as photosynthetic autotrophs. This is the assertion. Reason is because they contain chromatophores. Is it true or not? It is true because chromatophores contain chlorophyll A, and this chlorophyll A is responsible for it acts as the solar panel, and that is why cyanobacteria is able to synthesize its own food. Okay, very important, note down this. Shall we start writing the code words? Chromatophores are seen in where? Cyanobacteria. Or seen in cyanobacteria. Chromatophores are present in cyanobacteria, which is also called as a blue-green algae. Cyanobacteria is also called as a blue-green algae, but it is not an algae, it's a prokaryotic bacteria. Now, chromatophores, what are chromatophores? I am going to say they are plasma membrane extensions plus the pigments. What is the pigment? Chlorophyll A. Are you writing up? Are you writing up, students? If yes, please type it in the comment section. Yes. Okay. Now, chromatophores. Sorry, there is very less space. Chromatophores or write the code word plasma membrane extension. That extension contain what pigment? Pigment chlorophyll A. That is why they are called as photosynthetic autotrophs. This can be asked as an AR type of question. Usually what we expect? We expect like water chromatophores. Example of what is the other name for blue-green algae? We expect like this. But what if they ask like an AR type of question? Okay. So do concentrate on the students. Now, the third one is the main arena of cellular activity is cytoplasm. It is mentioned in NCERT. I strictly focus on NCERT. For the explanations alone, I'll go around and come back because NCERT is full of facts. You know. Today I'll share you the hack now itself because it's popping up in my mind right now. NCERT. Okay, this is how to study. I've seen lot of people, caring people who make videos like how to study NCERT. How to study NCERT. Students, there is technical glitch. Okay, for few seconds. So NCERT is full of facts, full of information. Okay, it's chunked. Now every word has to be decoded. Every word has to be decoded. It is like the DNA inside the nucleus. It is like the DNA inside the nucleus, which contains all the important info. All the important info. So what you are doing, you are studying the double standard. Okay, DNA, isn't it? You are decoding to have correct understanding about the DNA, isn't it? So same way NCRT has to be decoded. Every word in NCRT has to be decoded for better understanding. For better understanding, that is concepts. When you do decoding, you will be able to answer any question. I challenge you, you will be able to answer any question. See, I'll give you one word. Okay, decode it and tell me the answer tomorrow. Metabolic diversity. When I studied this word for the class, okay, there is metabolism is one word, diversity is one word. What is the meaning of metabolic diversity? That too. Now the question, where do you see metabolic diversity? If you know the answer, type it in the comments. Where do you see the metabolic diversity? In bacteria. This is a small prokaryote, right? This is a small prokaryote. How this can show such metabolic diversity? What is the relationship between metabolic diversity and bacteria? Find out, decode this and tell me. Okay, so NCRT, each and every word has to be decoded. Then you can easily answer all AR type of questions. Not only AR, any type of question you can easily answer. Okay, so now then one more thing I want to tell you. See, for cell chapter, we are doing it daily, right? But before, in the start, I was like, because of my cognitive disturbance, my mental issues disturbance, I was not able to do it as a whole. So I was finding lot of ways. To finish it off. Then I came to one way. That is, try this out. You can check it whether it works up for you. 1.5 pages per day means you can finish off cells chapter in totally around some 12 days. Just reading 1.25 pages per day, you can finish off the cell chapter thoroughly in 12 days. Okay, so try this and tell me how it works for you, how much you are able to complete in 12 days. Okay, next, coming back to this, that is, students, are you following me up? Okay, type S in the comments, students, so that it will be encouraging for me too. Okay, next, main arena of the cellular activities, cytoplasm. Cytoplasm is important. See, you have cell. You consider it as living and non-living. Now the Neet will check you. When do you say it as living? When do you say it as non-living? Suppose if you take off the endoplasmic reticulum, is it living or non-living? Suppose if you take off Golgi, take off nucleus from the cell, which is very, very important command center of the cell. Suppose I am going to remove nucleus from the cell. Do you say living or non-living? If you know the answer, type it in the comments. If you know the answer, suppose if there is a cell without nucleus, what do you call that cell? Cell without nucleus is called as enucleated cell. Enucleated, N-E. I am just pronouncing it as E, but you have to say enucleated cell. Can you give one example, students? Very, very easy example in physiology. If you know the answer, type it in the comments. If you know the answer is a cell without nucleus in blood is RBC, red blood corpuscles. Red blood corpuscles also called as erythrocytes. See, from cell we have dived into physiology. Okay, next, this is the one thing with me. I will just keep going and I'll end up in another chapter. Okay, so RBCs, these cells doesn't have nucleus. Do you mean that these RBCs are dead? RBCs are dead? Say yes or no. Type it in the comments, that is whether RBCs are dead or they are considered as alive. They are living cells. They don't have nucleus, Because of that, what will happen? They were not able to undergo cell division. RBCs cannot undergo cell division. What is the lifespan of RBC, students? Type it in the comments. Lifespan of RBC, type it in the comments. So it cannot undergo cell division. So that lifespan is short. Short lifespan. Okay, but they are considered as living. So if you remove the organelles, it doesn't mean that cell is dead. But if the cell doesn't contain cytoplasm, which expresses a living state of the cell, it is considered as non-living. Okay, so cytoplasm is the one which makes the cell a living or non-living one. Because what happens in cytoplasm? All the organelles, all the metabolic activities are taking place. So this is one question which can be asked in relationship to cytoplasm, can be asked as AR. Okay, whether living, how do you choose the cell as a living or non-living? The same example, RBC, because this example is given under nucleus. In the cell chapter, it is given it as under nucleus. Okay, RBCs are considered as non-living, or RBCs are considered as living because it does not contain nucleus. So what will be your answer? Type it in the comments, students. Type it in the comments. Next, see, this is the trap. Okay, Neet trap. That is, nucleus is absent, means is the cell dead? They can say that the cell is dead, but actually cell is not dead. Cell is considered as living. Cell is considered as living one. There is one question in NCRT next to that RBC and C tube elements. Go and check it. Okay, I framed this based on that only. So cell is considered as living. If the cell contains cytoplasm, then it is in the living state. It doesn't mind whether you remove out any of the organelles. Okay, the functions will be impaired. Okay, next is block 2, that is Golgi. Golgi apparatus has got 2 phases. How many phases? It has got 2 phases. 2 phases. Okay, one phase it is called as, first I'll read out, then I'll explain. Okay, cis phase and trans phase. Trans is the transport. Remember like this, trans is the transport. Cis phase, that is the coming in, entry gate. It is also called as the forming phase, which phase is the endoplasmic reticulum. Next, the other phase is the trans phase, which is the transport, that is exit gate, which is also called as the maturing phase, and it faces the plasma membrane. I will explain now. Okay, see, you have endoplasmic reticulum. Endoplasmic reticulum. Okay, this is the nucleus, and you have endoplasmic reticulum which extends. And close to this you will have, sorry, don't look at the diagram, this is just a pictorial representation. Okay, this is the Golgi, and see, this is the cell membrane, outer cell membrane. What is this space? Cytoplasm, the fluid matrix. Are you able to get? I am coming from the out to the in so that you can understand. You have cell membrane, then you have cytoplasm, then organelles are here. This is the nucleus. Now we are not going to worry about nucleus. We are going to study only this two. Relationship between endoplasmic reticulum and Golgi. Okay, this is the one which faces the endoplasmic reticulum. Here I have written, right, faces the endoplasmic reticulum. You call it as cis phase. You call it as cis phase, or the entry gate. From endoplasmic reticulum, the materials enter the Golgi. Here what happens? Now I am going to ask you the question. We already saw it. What is the function of Golgi? What is the function? Packaging and what? Modification. Packaging and modification of materials. What are those materials, students? What are those materials? Type it in the comments, students. They are glycoproteins and glycolipids. So this is like the factory which contains raw material. Endoplasmic reticulum is the factory which contains the raw material. From here, proteins and lipids are sent to the Golgi. Now the Golgi body will receive it through the trans phase. that is the coming in phase. It will receive it through the trans phase. Here it does the function, that is packaging and modification. This lipids and proteins are converted into, they are added a group and they are converted into glycolipids. This group is added. So now the lipids has become glycolipids, proteins has become glycoproteins. Now it will be sent out of the Golgi, either outside the cell membrane or to other organelle, any targeted organelle. Okay, it has got two pathway. So this modification happens here and it goes out. This transport phase is called as the trans phase. It is called as the trans phase. Very easy. The shall I tell you one example? Materials which needs to be packed, okay, packed and modified, enters from the endoplasmic reticulum. Think of this as a company which sends to this, okay, from here it is sent to Golgi Apparatus. Here what happens, packaging and modification is done. From there, that is the refined product is sent to the outside of cell membrane or to the targeted organelle. This phase which receives from the endoplasmic reticulum, that is the coming phase, is called as the cis phase. The phase where it is transported out is called as the trans phase or maturing phase. Okay, trans phase or maturing phase. Now I think you got it. This phase, trans phase, faces plasma membrane, cell membrane. Cis phase faces endoplasmic reticulum, right? So the code word is endoplasmic reticulum, cis phase or forming phase, Golgi, trans phase or maturing phase, plasma membrane. That's all. Students, did you get this? Or are you able to understand? Or am I too fast? Okay, I need your comments, students. I need your comments. Okay. Cis phase is the entry gate, that is the coming in phase, also called as forming phase, which faces endoplasmic reticulum. You can write it as ER. Okay, are you following me, students? Are you writing down? Cis phase is the entry phase or the coming phase towards the Golgi, also called as the forming phase, which faces the endoplasmic reticulum. Okay, now you have trans phase, which is the exit gate or the transport phase, also called as maturing phase. This is the plasma membrane. Trans phase of Golgi does the function of transport, that is it transports the packaged and modified glycoproteins and glycolipids. Okay, transports, also called as maturing phase, which faces the plasma membrane. Understood? Cis phase and trans phase. Cis phase, C for C, the phase which receives from the endoplasmic reticulum, also called as forming phase, which faces the endoplasmic reticulum. Next you have trans phase, also T for T, transport. It transports out, exit gate, also called as maturing phase, which faces the plasma membrane. Students, are you able to get it, students? Type it in the comments, students. We need to interact, right? Because I am focusing on the class. It's 30 minutes. It doesn't mean that you should not type. Okay, even when you are watching the replay, you can post your doubts, raise me a question and type the answers for the question which I have asked in the live session. And today block 3 is nucleus. Fleming, today's scientist, the hero today is Fleming. Can you remember the other scientist mentioned in NCERT related to nucleus? Who is that scientist? Robert Brown. N for N, Robert Brown. In the year, are you able to remember the year? Type it in the comments, students. 1831. If you know the answer, type it in the comments. 1831. So Fleming, after staining the nucleus using basic dyes, he named the material of the nucleus as chromatin. So if this can be asked as the match the following. Match they can give scientist and discovery. Okay, so Fleming, if you see Fleming, which answer you have to choose? Chromatin. Okay, Fleming, we'll write it one more time, only 3 times, right? Fleming, chromatin. There is no shortcut to success. Next is perinuclear space. What is the value of perinuclear space? You have to search for 10 to 50 nm. This is the code word, okay? 10 to 50 nm. What's nm stands for? Nanometer. Here they can make a trap. They can give you mm. So be relaxed when choosing the values. Be relaxed when choosing the values in the exam. I want to ask you one question. What are the breathing exercises you are all doing to keep you relaxed? When you do breathing exercise, okay, what nervous system gets activated? What nervous system gets activated? What are the 2 types of nervous system? Do you know the answer? Type it in the comments. You have sympathetic and parasympathetic. So when you do the breathing exercises, which nervous system gets activated and what nerve? I want the name of the nerve that makes you feel calm and relaxed. Okay. Next, perinuclear space. Value is 10 to 50 nm. Now you should question me, what is perinuclear space? What is perinuclear space? See, you have cell. This is the cell, okay, cell. This is nucleus. Now tell me, students, nucleus is a, how many layers, how many membranes? 2 membrane. Nucleus is a covering of made of 2 membranes. So it is a double membrane bound organelle. 2 layers, 1 layer and another 1 layer. Okay. So this is cytoplasm. Okay. So this one is outer layer. The inner one is inner layer or inner membrane. Okay, the covering of nucleus is 2 layered, that is double membrane bound organelle. The 2 layers are outer membrane and inner membrane. Together it is called as nuclear envelope. Nuclear envelope. You have seen envelope, right? That is a cover for the letter. So this acts as a cover for the nucleus. You call it as nuclear envelope. Nuclear envelope is made of how many membranes? 2 membranes, outer membrane and inner membrane. So in between the outer and inner membrane, there is a space, a gap. Space. You call that as perinuclear space. So perinuclear space is present between, tell me the answer now. If you know the answer, type it in the comments, students. Perinuclear space is present between the outer membrane and the inner membrane. Okay, this space is about 10 to 50 nanometer. Why is it called perinuclear space? Peri means surrounding. So surrounding the nucleus, there is a space. You call it as perinuclear space. Students, did you get this? Are you clear with the perinuclear space, students? I want some comments, some interaction, students. Even I will get bored if you don't comment. I want you to type locked. If you are able to recollect all this, I want you to type the word locked in the comments. Type the perinuclear space locked in your gray cells. So the nuclear envelope is the covering of the nucleus. What is the covering of the nucleus called? Nuclear envelope. What is the covering of the nucleus called? Nuclear envelope. The code word is nuclear envelope. Okay, what is the covering of the nucleus? Nuclear envelope, which is made up of 2 membranes. The outer membrane and inner membrane. Made up of how many membranes? 2 membranes. So it is a double membrane bound organelle. Are you writing, students? Double membrane bound organelles. The aim of this stream is to do active recall and active study, not a passive one. Okay, so I want you to write it down. Double membrane bound organelle. In between the 2 membranes, you have a space called as perinuclear space. You call it as Perinuclear space. Now the question for tomorrow is, what are the other organelles which contain space? Name the other organelles. Name the other organelles which contains space. Which contains space. Type the answer in the comment, students. Okay, one is nucleus. You have to find out the rest of the organelles. So the space between outer membrane and inner membrane is called as perinuclear space, which is 10 to 50 nm. Check this when you are choosing. Always when you choose the values, check the units next to that. Next, one important point. More than AR, it can be like statement questions. This can be statement question. Okay. This outer membrane is continuous with the endoplasmic reticulum, which contains ribosomes. So, which means it is rough endoplasmic reticulum. So it has got two membranes, outer membrane and inner membrane. Outer membrane is continuous with the endoplasmic reticulum. This is one important point. They will give you and ask you to check the facts about nucleus. Okay. See, I am showing here in the NCERT book itself. You have nucleus, this ball, nuclear membrane. See, this is the nucleus. They have marked pores, nuclear pores. Yesterday I asked you whether it is unidirectional or bidirectional transport, because from there should be communication between the nucleus inside the nucleus and the cytoplasm outside. Through this nuclear pores only the communication happens. Okay, now it has got two membranes. The outer membrane, see, it is in continuation with the endoplasmic reticulum. Now there is one question here. See, it comes now. The organelle which is close to the nucleus, which one is the rough endoplasmic reticulum? Now the next question, which organelle is close to endoplasmic reticulum? Just now we saw, we have Golgi apparatus. So name the four organelles of endomembrane system. If you know the answer, type it in the comments, students. Endomembrane system, nucleus, here you have endoplasmic reticulum, then Golgi bodies, then what? Lysosomes and vacuoles. Those four forms the endomembrane system. See, here it is given. Because their functions are coordinated, that is why they are placed close to each other. Easy way of communication. Okay, you have nucleus. This outer membrane extends with the endoplasmic reticulum. Okay, now this endoplasmic reticulum, ribosomes, which contains ribosomes, rough endoplasmic reticulum, it has got all the raw materials, that is the proteins. Okay, from there it is passed on to Golgi complex, where the modification happens. Then it is conveyed either to lysosomes or vacuoles, or if it is a complete product like insulin, it will be expelled out of the cell membrane, that is exocytosis, sent out of the cell for the use or function. Okay, now you understood, students. The outer membrane is continuous with the endoplasmic reticulum, which contains ribosome, which means it is the rough endoplasmic reticulum. Students, shall we do a recap? Are you able to remember, students? Are you able to understand these code words? Are you able to remember? If you want me to change anything, change the pattern, or you want me to be slow, I'll do all those things. We'll do fine refinements. Okay. So, a full recap. Under block 1, we saw polyribosomes or polysomes. There are several ribosomes attached to a single mRNA chain, which is called as polysomes. The function of polysomes is translation of mRNA to proteins. The polysomes are present in cytoplasm of the prokaryotes. Next is the chromatophores. They are the solar panel of cyanobacteria, which means these chromatophores are plasma membrane extensions which contains pigments, and the pigment is chlorophyll A. Okay, that is why cyanobacteria are able to synthesize their own food. They are called as photosynthetic autotrophs. Important AR type of question. Next is main arena of cellular activities. This can also be asked as an AR if they want you to check in a tricky way. Okay, cytoplasm is very important to consider a cell as a living or a non-living one. Okay, so because it is the place for all the main cellular metabolic activities. Next, if the nucleus is absent, the cell is not considered as dead. It is not able to undergo cell division and the cell will have a shorter lifespan. So RBC is a living cell. It's not a dead cell. This is the answer for the question in the NCERT. Next is block 2. You have Golgi, which has got two phases. We are going to see the two phases of Golgi. Okay, first is the cis phase, that is which is towards the endoplasmic reticulum. It is the entry gate, also called as the forming phase, which receives the material, okay, raw material, that is proteins. Next is the that is transports out, or also called as maturing phase, which faces the plasma membrane. Through this phase, maturing phase only, the finished product the finished product is sent out, the completed product is sent out. Okay, so this phase is the plasma membrane. Next, block 3, nucleus. Under this, we saw one scientist, very important, Fleming, in relation to chromatin. He is the one who gave the name chromatin after staining with basic dyes the material of the nucleus. Next is the perinuclear space. Before that, we will start this point. Okay, nuclear envelope. The covering of nucleus is called as nuclear envelope. This nuclear envelope is made up of two membranes, the outer membrane 1, inner membrane 2. In between these two membranes, you have what? Perinuclear space. This space is of about 10 to 50 nanometer. Check the units when you choose the question. This outer membrane is continuous with the endoplasmic reticulum, which contains ribosome, that is rough endoplasmic reticulum. Next, what is the function of nuclear envelope? It separates the material, that is the contents, between the cytoplasm and the nucleus. Okay, this around you don't need to study at all. You just need to remember. See, there is a separation, which means what? This should not get mixed up. That's all, very easy, right? So this nuclear membrane separates the contents from getting mixed up with the, that is, contents of nucleus from the cytoplasm. That is the function of what? Nuclear envelope. It separates the contents of nucleus from the cytoplasm. So what is the communication? I told you what is the communication between the nucleus with the surrounding, that is the cytoplasm and other organelles, through nuclear pores. Through nuclear pores. Okay, students, I hope you enjoyed and you logged all the vital information. I want you to type it in the comments, that is if you have understood, and if you have graces, I want you to type logged day 10. Okay, I want you to type logged day 10 even after the live stream ended, if you are watching the replay. Okay students, I am signing off with this today, and tomorrow we'll see with another interesting code words. Thank you students, thank you for listening and patiently writing with me. Hope you are writing with me, because we should be, if we make a commitment, we should be true to that commitment, which means we should be true to ourselves. Okay, students, stay tuned with Neet Cognition. Thank you.